Coming up on Dr. Kiki's Science Hour, we're talking about astrocytes in the brain with Dr. Cendra Agulhon from UNC Chapel Hill. That's up next on Dr. Kiki's Science Hour.
Netcasts you love.
From people you trust.
This is Twit. Bandwidth for Dr. Kiki's Science Hour is provided by Cachefly at CACHEFLY.com.
This is Dr. Kiki's Science Hour with Dr. Kiki, episode 75, recorded on December 2nd, 2010. We are made of stars. Welcome, everyone, to another episode of Dr. Kiki's Science Hour. I'm Dr. Kiki Sanford, and for the next 50 minutes or so, we're going to be talking about the brain. Now, when you think about the brain, you probably think neurons, right? Neurons are the things that make us act. They give us our behaviors. They allow us to think. They are the core of so much of what we do and who we are, right? Well, maybe they're not the only thing that has anything to do with who we are and what we do and how we do it. There are other cells in our brains that are really important, and researchers over the past several decades have been learning more and more about the complex and intricate environment that our brains are. And so today we're going to be talking about a group of cells that are known as glia, specifically within those, a group of cells called astrocytes. They're a type of glia. And now this year has been full of really interesting news regarding astrocytes. And here with me to speak about it today is Cendra Agulhon. She is a postdoctoral researcher associate within the UNC Chapel Hill School of Medicine's Department of Pharmacology. She works in the lab of Dr. Ken D. McCarthy, studying the role of astrocytes in neuronal signaling transmission, and is the first author on a paper that was published in the journal Science this spring that suggests that we don't know as much as we think we do about astrocytes. Cendra, welcome. Thank you for joining me.
Welcome.
Yes.
And thank you.
You're welcome. Now, I'd love to get this started with kind of a breakdown of astrocytes and glia and just kind of set the background for everything that led up to your paper this year and kind of set the scene for what's happening in the astrocyte world. So what are astrocytes exactly? What do they do?
Well, astrocytes are, like you said, a population of glial cells. And until a couple of decades ago, astrocytes were thought to be simply the glue that holds nerve cells together, insulate synapses, provide metabolic nutrients to neurons. But interest in astrocyte function has increased dramatically over a decade ago because of the newly reported roles. And among these roles, astrocytes have been found to be involved in synapse formation, control of vasculature tones, and also modulation of synaptic transmission and plasticity. Well, our lab and my research focus on this last role, the role of astrocyte in synaptic transmission and plasticity. And that has been a pretty hot topic in the role of astrocytes since, like I said, until a decade ago, astrocytes were thought to be passive cells, and neurons were thought to be the only cells that are active and process informations. So that basically the recent history view of the role of astrocytes. And then coming to our work and my work that has been published in Science in this year, beginning of the year, we actually provided new data showing that things are not that simple, and this role that was attributed to astrocyte and this role that showing that astrocytes might modulate synaptic transmission and plasticity, meaning might modulate and be involved in learning and memory, was not that simple. So I don't know, do you want me to go in more specific details about
Yeah, well, coming at it from the historical perspective is so we've over the years come up with this view that astrocytes are an integral part of the neuronal transmission, and that they play this really important role there. How do they how is that role based? What do they do exactly that why do we think that they are so integral to the transmission, or did we think?
Okay, so I can explain that. So basically what has been shown is like astrocytes express the same range of receptors to neurotransmitters as neurons do. And that was basically a very important discovery, and Ken McCarthy lab published a seminal work in the 19th showing that, meaning that these astrocytes could actually listen to neuronal conversations. And now it's very well established in acute slices, but also in vivo. What happens is these receptors in astrocytes that are called G protein coupled receptors, they are metabotropic receptors, can be activated by spillover of neurotransmitters from the synapse. And most of these, or not most, many of these GQ GPCRs signal through GQ, meaning that when these GQ GPCR receptors are activated, that results in calcium increases in astrocytes. And basically, these GQ GPCR calcium elevations in astrocytes are mainly considered a form of astrocytic excitability, and would be the primary link between neuronal activity and astrocytic excitability. So that was the first seminal work, and then after that, the neurobiologists became very excited, thinking that, well, if astrocytes can listen to neuronal activity and get these calcium increases, what does that do? Can they reply to the neurons? Can they modulate neuronal activity? And that has been under investigation over the past 18 years. And basically, researchers, including our lab, have used different pharmacological approaches to elevate calcium increases in astrocytes. And by doing that, by using pharmacological approaches that of course are not physiology, but that was a tool we had until now, we could see and we found that increasing calcium in astrocytes trigger release of neuroactive molecules that are called gliotransmitters, and that these molecules can modulate neuronal activity, synaptic transmission. And that was pretty exciting to us, because it was telling us that astrocytes might also have a role in synapse, in information processing and synaptic transmission.
Do the astrocytes I know that the neurons, their rate of firing is very quick. Are astrocytes in the same time scale? The calcium buildup and the release of the gliotransmitters, do they act in the same time scale that would really imply that they would have an effect?
Yeah, so that it's a very interesting question you're raising here. These GQ GPCRs are not ionic receptors. They are metabotropic receptors. So the calcium increases is it's long-lasting compared to neurons, even though it's still fast, but it's not the same time scales. And it's not, for example, in neurons, calcium increases in the presynaptic terminals that trigger presynaptic release of neurotransmitters are very fast, millisecond time scales. For astrocytes, it's different. These GQ GPCR calcium increases can take a second, several seconds. So the rise time and the decay time is lower than what occurred in neurons. So that means that, of course, it's not the same scale. So astrocytes are not neurons, of course. Yeah. And then, so that was basically the background I told you. But recently in our lab, because these this pharmacological approach that has been used so far, and we used that, and we found that neurons, astrocytes can release neurotransmitters have limitations. They are not cell-specific. So while we try to activate GPCR and astrocytes using ligands to endogenous GPCRs, we also activate all cells in the brain, because it is clearly established now that all cell types in the brain express an overlapping array of GPCRs. So that was a big limitation. And the second limitation, there are other pharmacological approaches that has been used, such as engaging calcium in single astrocytes, or poking an astrocyte with the tip of a pipette, or depolarizing astrocytes. Doesn't reflect physiology and do not recapitulate the full activation of a GPCRs. So in our lab, we took the approach in order to overcome these limitations to develop new tools, and we did that, and we developed new transgenic lines, mouse lines, that can allowed us to or selectively stimulate these GPCRs in astrocytes or block their activity. And by using these new tools, we actually could not reproduce our previous data and the data of the field, and we basically end up concluding that associated GPCR calcium signaling do not trigger gliotransmitter release and do not modulate synaptic transmission and plasticity. So that was the work you just mentioned before. So I was
just going to say, so it goes, the transgenic research that you've done is the complete opposite in its finding from the pharmacologic research that's been done previously. And so do you think it is a factor of the limitations that the pharmacology has? Like you mentioned that being not cell specific, you're adding things in to a dish and with cells in it. And so if you have only astrocytes, you're acting on astrocytes. Right. But if you have a combination of neurons and astrocytes and other cells, you're probably affecting all sorts of cellular behavior. How does the transgenic modulation, actually removing that calcium processing ability in the astrocytes, how do you think that specifically gets at the problem in a way that the pharmacology can't?
First of all, we selectively inactivate and obliterate GPCR calcium increases in astrocytes. Yeah. So all the other sources of calcium are maintained. We basically, in our mouse model, we have one mouse model that allows us to selectively stimulate a GPCR selectively in astrocyte, and another mouse model where we can block the calcium release due to GPCR activation selectively in astrocytes. So the differences between pharmacology is, first we touch to the endogenous regulator of astrocyte calcium, the GPCRs or the IP3 receptor. I haven't mentioned that, but the way to inactivate GPCR calcium increases in astrocytes is by knocking out the IP3 receptor at the membrane of endoplasmic reticulum.
So you're just knocking out the receptor. You said the IP3 receptor. It's a particular receptor that then affects calcium downstream. So if you knock that out, it knocks out the calcium response.
Yeah, exactly. Yeah, it knocks out the GPCR mediated IP3 dependent calcium release. And what I should add is we knock out the type 2 IP3 receptor that has been shown to be exclusively expressed in astrocytes, as opposed to type 1 and 3 that I found in neurons. So by knocking out the type 2, we knock out selectively calcium release due to GPCR activation in astrocyte. And we published that in Journal of Neuroscience in 2008 and found that these astrocytes in this knockout mice has no calcium increases. We can apply cocktail of ligands to different endogenous GPCRs and we don't trigger any calcium increases. So to answer your question, the difference between our model and what we previously used is that we really modulate or activate or block selectively the endogenous, the regulators of astrocytic calcium increases. We touch to the GPCR or the IP3 knockout, the IP3 receptor, sorry.
Right.
And we don't add calcium in cells. We don't poke an astrocyte that might mainly be reflecting pathology. So I think we believe that all models, of course, have limitations too, but are a major improvement over the pharmacological tools we have been using so far.
I'm wondering with the transgenic mice here, the question that often comes up with transgenic studies is, are you looking at adult mice, and how does the knocking out of this calcium modulation within the astrocytes specifically, because you're knocking it out only in astrocytes and not in neurons, how is that going to affect the development of the brain? And don't you think that through the development of the organism's lifetime, by the time you get to it, that some other that there might have been some kind of cellular modulation to make up for the fact that it's lacking that calcium channel?
Yeah, you're bringing a very important point, and what you're talking about would be a kind of compensation. When we knock out a receptor protein, we could think that the organism or the organs or the cells are going to compensate. Well, we actually tested for several kinds of compensation, and we did not find any. So we tested for the compensation for the overexpression of the type 1 and 3 IP3 receptors that could compensate the lack of type 2 in astrocytes. And there is no overexpression or expression of these receptors because we cannot trigger any calcium increases in any astrocyte we looked at. It's like a 100% knockout, at least in the hippocampus and cortex, where we looked at in details. And then in terms, there is other kind of compensation we can think about. It could be maybe the synapse can just adjust and start overexpressing postsynaptic receptors, AMPA receptors or NMDA receptors to compensate for this lack of gliotransmitters released from astrocytes. But so we have unpublished data that should be some work that should be submitted soon, but we found that there is no overexpression of AMPA or NMDA receptors. And we also are looking at the number of synapses, so that are undergoing studies, but so far we don't have any evidence that there are more synapses in this IP3 knockout. So again, that are the most, the first kind of compensations we can think about, and we looked at. We didn't find any compensation.
Okay. So you didn't seem to find any compensation for the lack of the gliotransmission, that the fact that the astrocytes weren't able to communicate in the way that they're normally thought to. So exactly. So I think it's just as research is, with new technologies, we can address new questions or find that what we previously thought was real is not that simple. So I guess with our new tools, even though there are limitations too, but there are an improvement for sure. So with this new tool, what we found is like things are not as simple we have been thinking, and so it just opens new avenues. We can maybe kind of rule out one possibilities, but then it opens new avenues for searching about what could be the role of these GPCR calcium increases in astrocytes. And that is very important because now it's under debate, right, since our work, but it's very important to understand that the role of these receptors, because
about 80% of neurotransmitter and hormones released in the brain act through activation of GPCRs. And approximately 50% of the therapeutic drugs on the market act on GPCR. So it's very likely that the therapeutic drugs used to treat neuronal disorders exerts their effect through activation of astrocytic GPCRs in addition of neuronal GPCRs. And yet, virtually nothing is known about the role of these receptors in astrocytes in normal brain function and as well in pathology. So I guess our work, even though we kind of rule out one possibility, I think what is important, it opens new possibilities to look at. Because we have been maybe focusing a lot on that glial transmission phenomenon, which is pretty exciting. And also what I think I should add is like, our work don't show that glial transmission does not occur, but it's shown that the phenomenon of the glial transmission concept as it is conceived, which means it's not happening like that, because this concept was activation of GPCR, calcium increases in astrocytes trigger gliotransmitter released. So if there is any gliotransmitter release, it seems that it's not GPCR calcium dependent, meaning it's not activated, it doesn't respond to neuronal activity. But it's very possible that astrocytes release gliotransmitters or neuroactive molecules in a homeostatic tonic manner, maintaining the homeostatic level or the ambient glutamate concentration or adenosine concentration in extracellularly. And that's very important too. But so I think it's just the way research goes, and there are so many questions now we can ask.
Yeah, well, it seems like the main point that's being brought up is that over the past couple of decades, the story that had been formulated for these astrocytes, the glial cells, and how neuronal transmission would affect them in a calcium-dependent manner by changing the amount of calcium in the neighborhood. The astrocytes would pick up the calcium, increase the calcium internally, and then spit out these gliotransmitters in response. That maybe there's something more subtle going on, that there's, like you said, that there's some other kind of, that the gliotransmitters are there. They're there because you've seen them. Researchers have found evidence for the gliotransmitters, but what is actually causing them to work, and how do they work? Why do they work? There's still all the nuances are not understood, and so your study, it's definitely something that has brought a lot of debate to the field.
It did. That's true. But like I said, it's just the way research goes, right? We have to ask some questions, use new technologies, and then rule out some possibilities and open new avenues for research, right? And like I said too, these gliotransmitters can be released, not necessarily in an activity-dependent manner, or not necessarily during physiology. It could be like during pathology. We found that in our lab, we published that if we get astrocyte swelling, but that happen in many kind of neurological disorders, astrocytes released glutamate and affect neuronal activity. So that still a possibility to investigate, and that's pretty interesting too, because we don't know also a lot about the role of astrocytes in pathology.
Yeah, and you mentioned in different drugs for different neuropathologies or for disorders, the idea that we use drugs that could affect neuronal function through the astrocytes, through an astrocytic pathway, but we never designed them to do that. We designed them for a completely different pathway, and then we might be finding out that they're working a little bit differently than we originally thought.
Yeah, exactly. And also that we are talking about astrocytes, but other glial cell populations express an overlapping area of GPCR. So the same therapeutic drugs can also act through activation of other glial cells. So, yeah, there is a lot. I think we are just seeing the tip of the iceberg, and there is a lot to learn more about glial cells in general and astrocytes in particular.
Yeah, in terms of their role in plasticity and in memory, you said that you've been looking at them specifically in the hippocampus, and that's the area that you've been focusing on. But astrocytes, they're all over the brain. They're not just in the hippocampus. Why did you focus specifically in that area, and why is their role in the plastic changeability of the brain, and which is inherent or necessary for memory to take place? But why did you look at them specifically there?
Yeah, this is a great question. Well, I did focus on hippocampus because first, hippocampus, it's the structure of the brain that has been clearly involved in learning and memory. And the second reason is because most of the studies looking at glial transmission has been performed using the hippocampus as a model system, basically the Schaeffer collateral C1 synapse as a model system. So in order to compare our data with previously published data, it was important to start to study a synapse that was already well described, and glial transmission were also found or reported at this synapse. So that was the two main reasons I focus on hippocampus.
Are you finding that aside from the calcium dependent nature of not showing the calcium dependent nature of the astrocytes now in their gliotransmission, what other pathways, or do you have any other ideas for how they could be involved in that region in memory formation?
You're talking about the calcium dependent, the calcium or that kind of pathway?
Yeah, Go ahead.
Other receptors, you're saying?
Not the receptors. I'm just curious about the astrocytes in general, since you're not seeing that they have this. You didn't show that they had the role that has been thought.
Yeah. So I think there is maybe two answers here. First, of course, astrocytes might have different function depending on brain areas. So now we have to look at other brain areas like visual cortex or somatosensory cortex, any kind of brain area that haven't been studied using our mouse model. So that's something we are going to do. I mean, we are actually doing right now. But then the second thing was astrocytes express transporters to neurotransmitters, and these transporters to neurotransmitter can maintain the good homeostatic level of glutamate after synaptic transmission. So these transporters are involved in synaptic transmission and plasticity. So that might be that is the role of astrocytes in synaptic transmission and plasticity, but much more passive, since they are taking up the spillover of neurotransmitters, and it's not something which is activity dependent. It's transporter just transport and take up glutamate, and the more they are glutamate ready, the more they take up, but it's not an activity dependent phenomenon.
Okay.
Did I answer your question?
Yeah. For the astrocytes, and I asked earlier about the time dependence of neurons versus astrocytes. Just out of curiosity, do you think in terms of our short-term versus long-term memories, there's often been a question of how do we remember things for years and years and years when we know that there is cellular turnover, cells die? How do we maintain those memories? How do they get put into place? Are astrocytes and their maybe longer, slower time scales of working potentially involved in the laying down of those really, really, I guess, durable memories?
Well, that's a very interesting question, and I don't think there is so far any evidence showing that, but it's possible. It's just because the studies are just starting about the role of astrocytes in memory and learning, but there is a possibility that this GPCR calcium increases, like it is the case in neurons, actually trigger mRNA transcription and protein synthesis that would be a long-term effect of activation of astrocytes. And then whatever protein would be synthesized might then have an indirect role in synaptic transmission and plasticity. But that is still has to be found or studied, but I don't think there is any data so far showing that. But that's something very possible. It could be that actually we are looking at short-term scale of astrocyte effect, but it might be, like you said, a very long-term effect.
Yeah. How about other cognitive functions? You said that different cells in different areas of the brain, that they have different functions. So an astrocyte in the hippocampus might have a different function in, say, the frontal cortex or the cerebellum. Are you looking or going to look at different areas of the brain to see whether or not your results pan out in a similar way?
Yeah, it's what I'm doing actually right now, because I'm doing two things, and I think that was important. It was first look at different brain areas. And so right now I'm looking at cortex and visual cortex in particular, and also to look at in vivo studies, because I don't know if we mentioned that, but my study has been done in slice, like acute slice, hippocampal slices. But so far there is no, evidence both against and for glial transmission needs to be verified in vivo. We cannot for sure say that astrocytes in acute slices works as they do in vivo. So what I'm doing right now, I'm looking at different brain areas, which is the visual cortex, and I'm working on awake mice, which to us it's something that is, and it's a better model in a way that is closer to physiology. So it's possible that we find something different, and it's exciting.
Yeah,
actually.
Well, it is exciting because you're really looking into new territory, looking into areas of the brain in a way that they haven't been looked at before. And I really think that looking at these questions surrounding astrocytes and other glial cells, the fact that our brain is not just neurons and we have all these other cells in the brain. When I took intro neurobiology, the glial cells, they're the support cells. They're the ones they have, they just keep the neurons happy. They make sure that they have, they produce ions or other things that the neurons need. And it was just this given that the only role that they had were there for packing. And somehow I had this image of astrocytes as kind of like those, the packing peanuts that you use for sending things to people without getting them damaged. But the story is so different now. And I think
yes, it is.
Yeah, go ahead. I just feel like the...
No, it is totally, and there is...
breaking up a little bit.
Okay, yeah, I can hear you. Yeah, there is no doubt that astrocytes are much more important than they were previously thought. And there are very interesting data from Ben Barrett's lab in Stanford University that's shown that astrocytes can release molecules that contribute and actually make synapses. Without these astrocytes and these molecules, there is no synapse formation, or a few synapses. It also found that these molecules can make a synapse functional versus a silent synapse. So that's very important. Another part of the field, which is very important, is seen that astrocytes can also release molecules that modulate vasculature or blood flow. And of course, that's very important function because neurons need to be fed, and so astrocytes can take up nutrients, metabolic nutrients from the vasculature and feed the neurons.
Yeah.
So there are much, I mean, that is part of the newly discoveries that has been reported over the past or 2 decades, and that makes by itself astrocytes extremely important in the brain function. And also, I don't think we mentioned that today, but there are in the mammalian brain and in human brain, they are the most abundant cell types. So they must be doing something important, or at least according to their number and the volume they occupy in the brain, they are a very important cell. And like I said, yet we don't know a lot about what they do.
Yeah, I think that's just fascinating to me. They're not as dynamic as a neuron. So because they don't have that vibrant dynamic profile, they've kind of gotten swept under the rug in the research story until recently, really. And the idea that they could have some impact through communication with the neurons to actually affect the way that neurons transmit information. Basically to say either slow down or speed up or, do your job a little better somehow. It's just a new way of thinking about the entire brain environment and how it's put together. Looking at it,
but what you're saying, it might be still possible, but longer time scale. Glial transmission, as it was conceived, was activity dependent, meaning neuronally activated or induced by neurons, and astrocytes will reply or respond back to the neurons very fast in less than a second for a second or two. And doesn't seem, at least within the hippocampus, it doesn't seem it is a case. It doesn't mean, again, it's not the case in other brain areas, but what you just mentioned before, it could be something that takes, it will not be that fast, it will be long time range role of astrocyte. And in this case, we should not call that glial transmission, or we have to find another term to describe the phenomenon. Glial transmission was basically homologous to neurotransmission. Neurotransmission is calcium-dependent release of neurotransmitter in a very fast time scale, and glial transmission was in a way homologous to that phenomenon. It was calcium-dependent release of glial transmitters, not as fast as neuron can do, but still in a short time scale, like a second or two. So now if this GPCR calcium increases trigger protein synthesis or something or enzyme phosphorylation, and the effect of this enzyme phosphorylation can actually occur after 2 seconds. Already it's not the glial transmission concept as it has been conceived. So, but we haven't found that anyway, because in our recording we of course record more than 1 second. We record for several minutes. So if there is such kind of GPCR calcium phenomenon dependent role of astrocyte on neuronal activity, it might be maybe an hour later or half an hour later, but that needs to be checked out, I guess.
Yeah, the duration sounds like one of the most interesting aspects of it, especially if you're... Oh, go ahead. I was just going to say, especially in areas of the brain where, maybe you're not working at an instantaneous time scale, but working on modulation that does need to take place over a much longer time scale. So maybe the neurons can modulate themselves through feedback mechanisms on a very quick scale, but then the astrocytes surrounding them modulate the overall activity over a longer term.
It's possible too, and again, we
you don't know yet.
We focus. The thing is, like in our field, we have been focusing on GPCR calcium increases because we have the technologies to measure calcium using calcium dyes and calcium imaging. But there's also different kind of GPCR GPCRs in astrocytes. Some of them are GS protein couples, some others are GI protein couples, and we don't know about anything about what. And this one might be the one we should study too. And again, when GPCR receptors are activated, there is calcium increases, but there are a large array of signaling and regulating molecules that are activated downstream of GPCR's activation. And the study of these signaling molecules has been neglected due to the fact that it's not possible to measure or to detect these signaling molecules, as it is the case with calcium and because we can use calcium imaging. So, I would say that as a take-home message that our data don't say that there is no glial transmission, but I think our data would suggest that the mechanisms of glial transmissions, as they have been conceived so far, needs to be reconsidered.
Are people receptive to that idea, or are there a lot of people really stuck on the idea that gliotransmission, as it's been conceived, is the only way, and we have to keep going down this path?
That's a tough question. I guess some researchers, they basically build their career on the study of glial transmission. So for them, it might be difficult to change their mind so I guess for this kind of researchers, they are not very happy with some of our findings. But for other researchers, they think that that's the way science goes. Concepts are made to be found that they are wrong years later, and then the way science go, right? We can approach the understanding of a mechanism with our human techniques or tools, but we will never really understand the real physiological mechanisms. So I think we just have to accept that findings and discoveries are based on our technologies, and they will improve or be found wrong or correct with the next generation of technologies. So I guess to answer your questions, some researchers are happy and some others are not, but that's just the way it is in research. So and again, like I said, and I really want to emphasize that we look at hippocampus, one synapse in the hippocampus for the two main reasons I gave you, but our data do not rule out the possibility that glial transmission does not occur in other brain structures, or that astrocytes have different functions depending on brain areas.
Right. So there might be different functions elsewhere.
Yeah, our data just cannot rule out the possibility that astrocytes have different functions in different brain areas. So now, like I said, we have to look at other brain areas and test for this glial transmission concept in other brain areas. And astrocytes also have been thought to be, like you said at school, when we have been taught about astrocytes, they are basically been described as a homogeneous cell populations, but that's not the case, and there are more and more evidence showing that astrocytes will be like neurons. There will be among neurons there are excitatory and inhibitory neurons, and among inhibitory neurons there are different kind of inhibitory neurons. Same thing for excitatory neurons, and it's very likely that it's the same case for astrocytes. The problem so far that astrocytes have been called astrocytes because they have expressed some cellular markers that are GFAP, for example, that stands for glial fibrillary proteins, and these proteins are expressed in astrocytes. So everything expressing GFAP is called astrocytes, but that doesn't tell us about other proteins receptors, and it's very likely that astrocytes in the same brain areas, even though astrocytes that are just neighbor, express different GPCRs. Some of them can express GI or GQ or GS. So that we are really at the very beginning of understanding the complexity of astrocytes population. And the more we will know about that, the more we can actually address the question whether these cells can have different roles in different brain areas or at different synapses.
Yeah, I think that's one of the most interesting aspects of this, is that the deeper you get, the deeper you realize you have to go.
The more you know, the more you realize you don't know anything.
Exactly. And it's just fascinating to me. I mean, the 90s were the decade of the brain, and there was so much discovered. And this past decade, there's been even more discovered about the brain, and we're constantly updating our hypotheses about how our mind works, our brain and our mind functions, how it's put together. And it's amazing to me that even now, as we're getting to some really incredible technologies that are allowing us to probe the brain even deeper and to clarify things ever more, that we still find we're coming up short, and that the technologies are still not allowing us to get to the point that we want to be. The fact that we still can't really identify an astrocyte. There's just saying there's, okay, all these cells that have this protein in it, we'll call you an astrocyte. But maybe there is another protein that we should be looking at as well that we're not. Or even in looking at concentrations, how low in concentration of calcium do we have to go in our monitoring? What really is a threshold for some of these cells? And also in gliotransmission, we're finding the transmitters, what we think are transmitters, but are there other compounds being released? Are we just not finding everything because we're not looking in the right places yet?
I agree. That's probably one of the most important questions now, is to know what kind of molecules, in addition of these gliotransmitters, astrocyte can release, and what could be their role during not only neuronal physiology in adult brain, but during development of the brain and during pathology. And we still, like I said, we are at the beginning of understanding the role of astrocytes in the brain, and that makes this study very exciting, because we have a lot to discover.
Yeah, are you finding that there is as you're looking at the technology that's available to you, and over the years that you've been studying these cells, and as you're looking at the brain, are you optimistic about the technologies that are coming that you haven't learned about yet, that will be developed and will help you look at the brain? Are you optimistic that you'll be able to answer some of these questions that are still outstanding?
Yeah, I think technology goes very, very fast. So there are researchers that really focus on developing new tools. And the thing for us, when we study astrocytes, we have to be, most of the researchers in neuroscience are neurocentric. They study neurons, right? So we have to find a way to use the new tools developed to study neurons, and then use them to study astrocytes. And sometimes it might not be as simple as it looks like, but there are many new tools like these new optogenetics models or tools that allow us to selectively activate and inactivate a cell in a very fast manner.
Yeah.
So that's something we are starting to develop in our lab. There are some light activated GPCRs that we can put in astrocytes and activate and inactivate then with light sources. That will be very helpful to study the timing of the role of astrocyte in a brain area, specific brain area in the brain. In the alive mice, because also, of course, I think now more and more people start working in vivo, and there are new technology like 2-photon Cassio imaging, and that are beginning more and more efficient to go deeper in the cortical layers, and that hopefully most of the lab will be able to get these technologies, but that's the best way to study the role of a cell in physiology, right? Because it's better to look at alive animal than working on an acute slice.
Yeah, any day. When you're looking at a slice, it's not necessarily behaving the way that a live animal would.
No.
Do you think are people starting to call themselves gliobiologists? You said that you call yourself a neurobiologist or a neurologist, or there are these terms that reflect neurons. But as people are specializing more and more into this other world of the brain, are people starting to specialize out? And
I guess most of the people are still calling themselves neurobiologists, but gliobiologists, it's a term that is used. I don't know if it's more and more, but I guess it's used, and sometimes I call myself a gliobiologist. So it's just, neurobiologists, now that we know that glial cells made up most of the brain cells should not be called neurobiology, right? We should be calling us brain cells biologists.
Right. So that the word would actually represent that there are a lot more cells in the brain than just the neurons, right?
Yeah, exactly.
We're being a little biased, focusing only on the neurons. Let's spread out, people. There's a lot more to work on there. In terms of, we're coming to the end of the show, so I just wanted to get at your own personal interest in this subject. What drove you to study astrocytes specifically, but and then more generally the brain and the memory areas of the brain?
Well, that's also a tough question. Personally, I've never decided I would be doing that. I guess it's more I study, and my path in research brought me to these astrocytes in the brain. But I guess more generally speaking, I became captivated by the brain because I think it's a magic organ. Like it reflects so much of our personalities and behavior, and it's something very mysterious and magic. So I guess it was my way to kind of approach a little bit the magic of the human being, without knowing that we are very, very far from understanding this magic, and I don't think we want to understand the magic entirely. I think it's nice we don't understand everything anyway. But I guess might have been why I decided to study the brain. And then for the glial study, I guess I arrived at a little bit by accident, and I might have also been fascinated by what we just described before. These cells are so numerous and nobody was studying then, and when they must be doing something. So I guess to me it was interesting to study something that everybody was neglecting. So that was, I guess, maybe personality things, but I liked the idea to study something that is still new and untouched almost, but even though now there are more and more people studying astrocytes.
Do you see the field going in really exciting directions? Are you is this a field that you are excited to remain a part of? I think I will be working on this field for a long time, because like I said, we are just starting to understand the role of these glial cells, and in particular astrocytes, and I will also be working in plasticity and learning and memory, because I like this. I think it's still magic. So if I can understand a little bit of the magic, I'll be happy. I like that. the idea of magic and what makes us up, and trying to figure out the little pieces, trying to get some idea of what that magic is made up of. But like you said, it's always good to have a little magic left over that we don't understand.
Yeah. We don't want to understand everything.
Yeah, exactly. Thank you so much, Dr. Agulhon. It's been wonderful talking with you. I really appreciate your time this evening, and the work that you've been doing to help uncover the magic in our brains by looking at little cells called astrocytes.
Thank you for inviting me. It was very nice to talk to you.
You're welcome. You're welcome. Good luck with your research. I do hope that I will be looking for more studies to see what you're up to. So thank you very much.
I keep you posted.
Okay, wonderful. All right. Well, have a good evening. Bye. And that was Dr. Cendra Agulhon, and she is a research associate in the University of North Carolina, UNC, Chapel Hill. She works in the laboratory of Dr. Ken D. McCarthy, and if you are looking for more information on astrocytes and astrocyte signaling, you can go to their website. The website is www, it's kind of long, so hopefully we'll have a link to it on our website, twit.tv/Kiki. But the website that you can check it out at is med.unc.edu/pharm, PHARM/people/primary faculty /Ken -D-McCarthy-1/McCarthy-astrocyte-signaling. That was a mouthful, wasn't it? That was quite a mouthful. But there are some very interesting, an overview of astrocyte biology, their signaling, how they're approaching it in the McCarthy lab, what research Dr. Agulhon is working on, and some molecular studies that are going on as well. So it's a really interesting overview. There are some other stories available on the web that I found that are quite interesting as well. One that overview this entire debate. There's a great story in Nature News, nature.com/news. It's Neuroscience Settling the Great Glia Debate. Brings up all sorts of sides in this story. Additionally, another story on C&EN, which is the Chemical and Engineering News website. They have a story on questions arising over the interaction between brain cells. The original study was published in Science magazine. Hippocampal short and long-term plasticity are not modulated by astrocyte calcium signaling. This headline, the abstract, the study itself, making waves in the gliobiology world, suggesting that gliotransmission does not occur in the way, at least in the hippocampus, in the way that decades of research suggests that it did. So the conversation is definitely going strong in the neurobiology community as to what's up with the astrocytes. That's what researchers were looking at. And as Dr. Agulhon mentioned, it's the questions of how this is going to affect the neuronal disorders. How are our astrocytes involved? How are they evolved in many disorders of the brain? And also the drugs that we take to treat lots of disorders. Finding out that those drugs actually interact with the astrocytes as well as the neurons. And what's going on, we don't really know. There's so much we don't understand. So the stars in our brains, the astrocytes, they're an interesting area of study. There's some technical stuff going on, and the deeper the researchers go down the rabbit hole, the more we will find out and the more we will understand. But as she said at the end there, it's always good to leave a little magic left over. So with that, I will end this episode of Dr. Kiki Science Hour. Thank you very much for watching and paying attention this hour. I hope you did learn something. That's what I always try to do here. Educate you just a little bit, or at least leave you asking questions. That's what we like to do. You can subscribe to Dr. Kiki's Science Hour at twit.tv/Kiki and get all sorts of past episodes. It's great. You go to the website, twit.tv/Kiki. All the past episodes of the show. This episode will be there as well. You can subscribe via iTunes or any other catcher that you have that you enjoy. You can get it in audio or video. Both are available to you. Subscribe. We kind of like it when you do that. And additionally, you can follow my other sciencey pursuits online. Google Dr. Kiki. I'm at Dr. Kiki on Twitter. I'm also, I have a Dr. Kiki Facebook fan page. And you can, what else do I want you to do? Oh, yeah. Check out This Week in Science and Green Tech Today, my other shows here on the Twit Network. And that's it. I'll see you next week. Next week, we're going to be talking about robots in space. Not pigs in space, but robots. We're going to be talking with the lead engineer for R2. It's a robot going up on a space shuttle. One of the last shuttles going up. And hopefully we'll be talking to R2 too. That should be pretty fun. So until then, just remember, I only ask 1 hour a week. And isn't that enough to make your world a whole lot more interesting?